Hello, everyone, and welcome back to Bio 181. In this lecture, we will discuss meiosis and sexual reproduction. This lecture is going to use and build upon what was covered in Chapter 10, so it would be helpful to review your notes from Chapter 10 before beginning Chapter 11. In the previous chapter, we discussed how cells most commonly replicate themselves through the process of mitosis. These are cells that do not do sexual reproduction, like skin, heart, and brain cells. They are called somatic cells from the Latin root word for of the body. Checkpoint 1. In a human skin cell undergoing mitosis, how many chromosomes does the parent cell have? How many daughter cells are produced? And how many chromosomes do each daughter cell have? The cells that do not reproduce via mitosis are the gametes, sperm and eggs. They do not make daughter cells that are genetically identical to the parent cell. They are referred to as germline cells and are used by sexually reproducing organisms to generate offspring. These gametes combine together to form a single cell. As we are now aware, human cells have 23 pairs of chromosomes for a total of 46 chromosomes. Therefore, each gamete has half as many chromosomes. To put it another way, they have 23 unpaired chromosomes, or no homologous chromosomes. When an egg and sperm combine, they become whole again with 23 paired chromosomes, 46 chromosomes altogether. Because these gametes only have 23 chromosomes total, they must go through a different process of creation than mitosis, which would have created cells with full sets of chromosomes. Now would be a good time to refresh terminology. Remember that diploid cells describe cells that have pairs of what we call homologous chromosomes, di meaning two, and ploidy meaning chromosomes. Previously, we used the analogy of having 23 different books and two copies of each book. So diploid human cells have 46 chromosomes. Haploid comes from the root words half and chromosomes, have half as many copies. In other words, these cells have a single copy of each book for a total of only 23 chromosomes. Meiosis is cellular reproduction that generates haploid gametes, and germline cells are the special diploid cells that divide to produce the said haploid gametes. The image shown is a rough outline of this process. We'll revisit this image again in more detail, so don't become overwhelmed just yet. This process should look different to you than mitosis. At the top are primary sex cells, and they are shown as having diploid homologous chromosomes in sister chromatid form. Meiosis I separates the homologous chromosomes into separate cells. We went from one diploid cell to now having two haploid cells. Then meiosis II pulls apart the sister chromatids and creates four haploid cells in total. Checkpoint 2. Mitosis and meiosis both involve the same basic sequence of phases. What are the four phases of mitosis and what are they followed by? Unlike mitosis, meiosis has two cellular divisions called meiosis I and meiosis II. At the top are primary sex cells. By now you should quickly be able to tell we are looking at sister chromatids in that cell. There are two homologous chromosomes, a pair at the top and a pair at the bottom of the cell. Meiosis I separates the homologous chromosomes into separate cells. The sister chromatids are still intact at this point. We went from one diploid cell at the top to now having two haploid cells after meiosis I. Then meiosis II pulls apart the sister chromatids and creates four haploid cells in total. We can use jackjumper ants to help simplify the process. These ants have just one single pair of homologous chromosomes. In our analogy, they have one book and two copies. Each copy is color coded, so we can keep track of that one and the difference between the two being one from the mother and one from the father. Now, before we can jump right into meiosis, no pun intended, just like with mitosis, we need to replicate our DNA in S phase. So let's review that process now. Before S phase, that is how the chromosomes look. The cell has two chromosomes, one from the male parent and one from the female parent. After S phase, the cell replicates each of the chromosomes to make sister chromatids. At this point, there's a pair of homologous chromosomes and four sister chromatids. And this image has everything labeled start to finish in S phase. The cell replicates the DNA of each chromosome to make a sister chromatid. What confuses students here the most about S phase is that this does not, I repeat, does not change the number of homologous chromosomes. Remember, a chromosome is a distinct chunk of DNA. Because the newly made copy of DNA is still bound by the centromere to the original, it is still a single chromosome. So, at the top, we can count two chromosomes before S phase and two chromosomes after S phase. We instead made four sister chromatids that are attached by the centromere. And we have each one of the sister chromatids listed at the bottom. The DNA does not quite look as neat in S phase, though. The germline cell has a single intact nucleus containing two copies of DNA, but the DNA is in the form of chromatin. So, in prophase 1, named so because this is meiosis I, DNA condenses and winds into chromosome form. The nuclear envelope breaks down so that the chromosomes are mobile. And if you look in the polar regions of the cell, we see centrioles. Centrioles are cylindrical organelles that begin to build the spindle fibers and stretch them across the cell. Furthermore, homologous chromosomes physically pair up with one another in a process called synapsis. Now, because of their close proximity, they engage in genetic recombination, known as crossing over between homologous chromosomes. This process does not occur in mitosis. Mitosis generates nearly exact daughter cells, and crossing over would fundamentally change the daughter cells. Crossing over is the process of exchange of segments of genetic material from one specific location of homologous chromosomes to the other chromosome. The sites at which crossing over occurs are called chiasmata. In metaphase 1, pairs of homologous chromosomes line up at the equator of the cell while still in synapsis. In anaphase 1, the homologous chromosomes are pulled apart. The key here is the homologous chromosomes are pulled apart. In mitosis, we separated the sister chromatids in anaphase. Each end of the cell now will have a copy of the chromosome. In telophase 1, the chromosomes are clustered at opposing ends of the cell. A nuclear envelope forms around each one. So what's the difference here? From the first homologous chromosomes have swapped some of their sections of DNA. But second, these new nucleuses are not diploid. Now they are haploid. This is the result of anaphase. During anaphase in meiosis, the homologous chromosomes moved apart. And now the chromosomes decondense back into the form of chromatin. Finally, at the end of meiosis I, cytokinesis occurs. Remember, these new cells have a different chromosomal structure due to crossover, so the cells are not exact copies of the parental cell. So, we have now covered meiosis I, which resulted in the crossover and separation of homologous chromosomes into separate cells, called haploid cells. Now let's cover meiosis II, which, similar to mitosis, results in breaking apart the sister chromatids. Meiosis II begins with prophase. It's called prophase II. And no crossover occurs in meiosis II, as, this, as the cell is without its homologous chromosome. In metaphase II, the chromosomes align at the equator. The sister chromatids are separated at the kinetic core in anaphase II and pulled apart to opposite ends of the cell. And telophase II follows, and now we have a new pair of nucleuses. They no longer contain sister chromatids as in telophase I. And cytokinesis results in a total of 4 cells because we had 2 cells from meiosis I and they both divide into another set of 2 cells. So from the single diploid cell, we now have 4 offspring generated. The offspring have different combinations of genes on the chromosomes due to crossover, and moreover, they are all haploid. At this point, we should now recognize we have created gametes. Checkpoint 3. The somatic cells of domestic dogs have 78 chromosomes. How many chromosomes are found in dog gametes? Checkpoint 4. Before a human germ cell undergoes meiosis, is that cell diploid or haploid? Are the 4 daughter cells that result from meiosis II diploid or haploid? Now let's go back through meiosis I and meiosis II to be sure we can keep track of the chromosomes. Look at the initial cell. Before we begin, see if you can tell how many sets of homologous chromosomes and what ploidy the cell has. If you said the cell has 2 sets of homologous chromosomes and is diploid, you would be correct. After S phase and its DNA replication, how many sets of homologous chromosomes are there? There are still 2 sets of homologous chromosomes. It's still diploid, but we now have created the sister chromatids. After meiosis I, how many cells are there, and what is the state of the chromosomes? There are 2 daughter cells. Their chromosomes have gone through crossover, so they are not exactly as before, and each cell now only has 1 of each chromosome. They no longer have a set of diploid chromosomes for each, and are therefore haploid with sister chromatids. Finally, after meiosis II, how many cells are there, and what is the state of the chromosomes? There are 4 cells, and each cell has a single copy of each chromosome, and the sister chromatids have been separated into their own cells. And this slide right here kind of captures everything all together. So on the left side, we have mitosis, and on the right side, we have meiosis, which includes meiosis I and then meiosis II. Now let's compare side by side mitosis and meiosis I. We can see with mitosis in prophase, we get the separation of, we have their sister chromatids, and then they um, get brought into the same plane in metaphase. And then the sister chromatids are separated in anaphase. And in the process of the separation in anaphase, um, each set of homologous chromosomes both end up on both ends. So there's still a diploid set when we reach telophase. If we actually looked in telophase, there's two copies of each one of those chromosomes. All we did was separate the sister chromatid replicants. If we compare this over to prophase 1, the homologous chromosomes are brought together for synapsis, and then we get crossover that occurs. In metaphase 1, they align along the center axis there. And then in anaphase, the homologous chromosomes are separated from each other, not the sister chromatids like in anaphase and mitosis. Then in telophase, we have the formation of the nuclei around the newly formed or the newly separated chromosomes, and cytokinesis follows. Now let's compare meiosis II to mitosis. Now the set of steps here are very similar, but one thing you'll note in prophase of mitosis versus prophase 2 of meiosis is that these cells in prophase mitosis are diploid, and in prophase 2 they're haploid, because we separated the two cells, homologous chromosomes, apart from one another. In metaphase, they both align in the center there. And then in anaphase, we separate the sister chromatids. And then in telophase, we form new nuclei around each one of them. And then we go through cytokinesis or cell division again. The end result in mitosis, we have cells just like in the very beginning, the very starting cell. We have diploid cells, and they're not in sister chromatid form, but they have all of their sets of chromosomes in diploid form. However, if we look at meiosis II, the last phase there, we have haploid cells. There's 4 of them, and they have each one of their chromosomes, but they don't have homologous copies. Checkpoint 5. For prophase, metaphase, anaphase, and telophase, name everything that is different between mitosis and meiosis that was discussed. Okay, so why do gametes exhibit crossing over? In part, crossing over allows for more genetic diversity. We previously discussed how one homologous chromosome comes from the mother and one from the father, and they are like two versions of the same book. The stories or genes have some variations between each. By doing crossover, we can create books or chromosomes with new combinations and therefore greater diversity. In the case of mitosis, the goal is to replicate with a certain degree of fidelity or replicability between each. We need to have some consistency in our cells in order to have a certain order and continuity in the process and capability of the cells. So it's important that somatic cells do mitosis. Errors can emerge. Aneuploidy is an improper number of chromosomes. Looking at this karyotype, we can see that this person has triploidy for chromosome 18. This can happen when homologous chromosomes fail to separate properly in anaphase 1, or when sister chromatids fail to separate in anaphase 2. This is called non-disjunction. In metaphase, the chromosomes line up, but improper separation occurs in anaphase. And almost all somatic chromosomal aneuploidies are fatal before birth in humans. Aneuploidy is the most common cause of miscarriage. Down syndrome is the only instance where a human can live functionally for many years with three copies of a chromosome. This is the case because chromosome 21 is the smallest chromosome, carries the least amount of genetic information, and therefore having three copies of that chromosome has the least impact of any chromosome. Aneuploidies of the sex chromosomes X and Y include Klinefelter syndrome, where the sex chromosomes are XXY and results in less body hair, poor muscle development, greater height, poor coordination, abnormal breast growth, and speech and reading difficulties. Individuals with Turner syndrome have an X chromosome but lack a homologous sex chromosome to match it. They may have webbed neck, inability to have children, vision and hearing problems, heart defects, and thyroid hormone problems. Triple X, XXX chromosomal structures results in a normal female, and XYY results in a normal male, but YY and Y are lethal combinations. What does it mean to be intersex? 1 to 2% of the population is intersex. Intersex individuals have some combination of male and female sex chromosomes, ovaries, testicles, genitals, and/or sex hormones. Klinefelter syndrome is XXY aneuploidy, and ovotesticular disorders of sex development can involve eggs being fertilized by two different sperm or embryos or fraternal twins fuse to become one. And it results in with individuals with both XY and XX cells in the body. Androgen insensitivity syndrome XY occurs when XY individuals whose cells do not respond to testosterone and develop female or partially male characteristics. As women age, their germ cells undergo more non-disjunction events. Women in their 20s have a 2 to 3% chance of having a child with a chromosomal aneuploidy. Women in their 40s have a 30% chance of having a child with a chromosomal aneuploidy, and it's yet unknown whether this correlation is the same for men. However, in 2017, researchers in Iceland determined that older fathers pass on 80% more non chromosomal mutations than older mothers. And that's the end of the chapter. Thank you so much for joining us, and we'll see you in the next one.